Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about kinases and the phosphorylation of proteins, particularly in signal transduction cascades. So in our previous video about signal transduction and the basics of signal transduction, we indicated that engagement between a signaling molecule and its receptor on the target cell is going to produce some sort of intracellular effect, and very often how that's going to happen is through the activation or deactivation of proteins that are inside the cell. Now, before we actually go any further into the particulars of signal transduction cascades, like those you see with G protein coupled receptors or tyrosine kinase receptors, I wanted to dedicate this video to just the basic concept of protein phosphorylation, dephosphorylation, and how that affects protein function. So what we're going to see when we talk about all these different signal transduction cascades as we go throughout the course is that many of these pathways are going to lead to the activation of a class of enzymes called kinases. So we'll explore what kinases are here in just a minute, but like I said, since we're going to see this so very frequently, it's going to be worth our time to kind of dedicate a whole video to talking about it now. A kinase is an enzyme that transfers a phosphate functional group from one substrate to another. So in this case, you can see we have two substrates, a green one which is attached to a phosphate group, and a blue one which is not. The kinase is going to transfer that phosphate from one substrate to another. So in most physiological scenarios that we're going to be examining, the phosphate donor is going to be a molecule of ATP. Again, another very good reason why cells need to obtain as much oxygen as they can so that they can make a bunch of ATP through cellular respiration. You want to have ATP on hand to be a phosphate donor for these types of enzymatic reactions. So the ATP molecule is going to donate the phosphate, and the recipient is usually going to be some intracellular protein. Now, if you remember from, say, a general biology class, all the steps of glycolysis in cellular respiration, you're going to remember that a lot of glycolytic enzymes are also kinases. Those kinases transfer phosphates from non-protein substrates to other non-protein substrates. So this class of kinase enzymes that transfer phosphates onto proteins specifically are called protein kinases, and that is meant to distinguish them from the more kind of metabolic kinases. Now, in terms of protein phosphorylation, the phosphate is going to be attached to amino acid side chains. So this is going to take us back to a much earlier video. It turns out there are only 3 amino acid side chains that can potentially be phosphorylated. These are serine, threonine, and tyrosine. The thing you'll notice that these side chains all have in common with each other is a free hydroxyl functional group, and that is exactly where the attachment point for the phosphate is. So if a protein or a peptide does not have a serine, a threonine, or a tyrosine, that protein or peptide cannot be phosphorylated. So that's something to keep in mind. Okay, but what's the big deal with protein phosphorylation? What does it matter, right? So a phosphate group is teeny tiny in the grand scheme of how big the protein is. Why does phosphorylation of a protein matter? Well, hopefully you remember from a much earlier video that it is the folded three-dimensional structure of a protein that ultimately determines what that protein's function is. So in this protein, however it is folding here, that is going to produce some function for that protein that we'll call function A. So if we were to phosphorylate a protein, this very often stimulates a sometimes subtle, sometimes not so subtle shape change within the protein. These shape changes are often called conformation changes. So what you can see here is that the phosphorylation of a serine, threonine, or tyrosine residue within this protein has caused a somewhat minor shape change, although the shape change is definitely noticeable when you compare the two proteins to each other. So since this protein that is now phosphorylated has taken on a new shape, we can say pretty confidently it is going to have a different function now, which we will call function B. So, through signal transduction, if we activate a kinase that phosphorylates a protein, we can potentially change that protein's function, and the cell's response to the signaling molecule is going to probably be in large part due to the change in function for this protein or some set of proteins that are found inside the cell. So don't walk away from this thinking that phosphorylation is a minor thing. It is actually a very major thing because it can completely turn around how a protein functions inside the cell. So the takeaway here is that even if you just change the shape of the protein a little, that can actually produce a very large and very noticeable change in effect on that protein's function. If the shape changes, the protein's function is going to change as well. So the idea here is that phosphorylation is basically acting kind of like a switch. The cell, through activation or deactivation of kinase enzymes, can flip these switches on and off at will. So what you can see in this illustration is that a protein here, and this is just a fictional, or not a fictional scenario, but this is just kind of a hypothetical scenario right here. This protein here, which is not currently phosphorylated, is inactive. So this protein is not doing anything. It exists, it's floating around inside the cell, but it's not doing anything, it's inactive. So if we were to activate a kinase enzyme so that we phosphorylate this protein is going to take on a conformation change. It's going to change its shape, and now this protein will actually be active, and it will start doing some task or doing some job that it was not doing previously. So, like I said, it's like flipping a switch on and off. The cell, through molecular control of kinase activity, can determine when proteins are active and when they are not active based on physiological conditions that cells in the body are going to communicate to each other through neurotransmitter release or through hormone release. So again, I mentioned this before, kinases are going to be very common targets of signal transduction pathways. Once a ligand binds to its receptor, you can potentially activate a kinase enzyme, which will then start adding phosphates to a bunch of proteins inside the cell, and in doing so, either turn those proteins on if they were previously off, or turn those proteins off if they were previously on. But the idea here is that through phosphorylating a target or effector protein, the kinase has the ability to change that protein's function because the protein shape is changing. So let's illustrate what I was just talking about. In the absence of a ligand, you will often have a kinase that is inactive in close proximity to the receptor. If there's no ligand bound to the receptor, then the kinase will be inactive, and any intracellular proteins that are targets of that kinase will also be inactive. But once the ligand binds, the receptor transduces the signal to the kinase, and the kinase now becomes active, and the kinase will then add a phosphate onto that protein and activate that protein as well. And then the rest of the intracellular physiological response will be owed in large part due to the activity of whatever this protein is going to do now. Okay, so one last thing to cover here. So it is true that the function of kinases is to add phosphates to proteins. However, we don't want to give the kinases too much credit. While kinases can add phosphates to proteins, they cannot remove them. So this is very much an irreversible reaction as far as the kinases are concerned. So that's going to make you wonder, okay, well, if this protein here is inactive and we phosphorylate it with a kinase, does that mean this protein is now permanently active? Is there any way to get this protein to be inactive again? And the answer is yes, there is, but it's going to require a different kind of enzyme. The enzymes that remove the phosphates are called phosphatases. So kinases add the phosphates and phosphatases remove them. So you're actually going to probably see a couple of examples of this throughout the course, but phosphatases are actually subject to the same kinds of regulations as kinases. A signal transduction pathway could activate a kinase enzyme to phosphorylate a protein to change its function in that way, or a signal transduction pathway could also just as easily activate a phosphatase enzyme to remove the phosphates off of proteins inside the cell. Either way, by toggling a protein between its dephosphorylated state and its phosphorylated state, we can still flip that switch back and forth. The kinase enzyme will flip the switch one way, the phosphatase enzyme will flip the switch the other way. Okay, that's going to pretty much do it for this video. Here is a list of vocabulary terms you probably ought to be aware of. And as for checking your understanding, number 1, how many times can the peptide that has the primary sequence MLK, YPATE, SGACR be phosphorylated? Number 2, how could a kinase enzyme cause the functions of other proteins inside the cell to change? And number 3, can kinases catalyze the reverse reaction, i.e. the removal of a phosphate? Okay, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section, and I will see you next time. So long.